So we're looking at cell signaling, and we've already talked about the types of signaling. Now let's look at some of the signals themselves. So we could have what are known as hydrophobic signals. And hydrophobic signals, because they are hydrophobic, it allows them to easily pass through the membrane. So the hydrophobic signal can come through the membrane, and as it comes through the membrane, it interacts with the machinery inside. And what this is going to do is it's going to allow for incorporation and gene transcription. So it's going to make its way into the nucleus to allow to become involved in gene transcription of DNA as well and incorporation of DNA. An example of this is a steroid hormone. As we know, steroids are hydrophobic, and so it's a good example. And so we also have hydrophilic, and so the hydrophilic compounds, when they're trying to make their way through this membrane right here, because it's a lipid bilayer, it's a little more challenging. So they're going to need receptors to recognize the signal and allow for the signal to pass through. So if we imagine this right here is the signal, what's going to happen is you have something like insulin, and it's going to come to the receptor, and that receptor is going to send in a signal inside the cell and tell the nucleus what needs to be done and allow for gene transcription. So that's the difference between your hydrophobic and hydrophilic signals. So remember, your steroid is your hydrophobic, while your insulin is your hydrophilic. And this is the signal. And so typically, when we think of external stimulus or signaling, what's really happening is we have an external stimulus, and this external stimulus is going to send information to the decision maker. And basically what that means is the integration center. And then once it sends it to the integration center, it's going to result in a response. So if we think about some external stimulus, like putting your hand on the stove, that's more of a macro than a micro. The external stimulus being the heat that is going to the sense receptors on your hand. The signal is going to go to your brain, make the decision, and then there's going to be a response to pull your hand away. This is kind of a similar thing. Now we're looking at, on the micro scale, what neurotransmitters are released, what happens between the different signaling patterns. Are we going to have neurocrine signaling patterns, paracrine, endocrine, or autocrine signaling patterns to control that response? Now, if there is some sort of issue in the decision-making center, so we have an abnormal signaling or an abnormal signal transduction. So if this is abnormal, that basically can lead us to cancers or other human diseases. And ultimately, if we're looking at this process itself as discussed, this process that we see right here under normal situations basically leads us to controlling cell function. And that includes metabolism, secretion, contraction, proliferation, differentiation, transformation, cell survival, as well as cell death. In this, we also have different types of communication within the cell. So if we simply look at what are known as gap junctions, we have two cells that come into proximity, and they have these, you can almost look at them as bridges that can allow for communication to occur, and these bridges are your gap junctions. And the gap junctions are made of connexons, and each individual connexon is made by a connexin. So I know it sounds a little confusing, but just remember, if you see connexins and connexons, we're relating to gap junctions. And basically our gap junctions are used for sending electrical signals and they allow for rapid transmission of action potentials. They're also going to be diffusing metabolites and second messengers. So this is the important thing about gap junctions. Now, we also have contact-dependent signaling, which means that when we come into proximity of something else, once we contact the target, we're going to have a signaling pattern. And so basically, the molecule binds to the plasma membrane, and it's a direct interaction with the bound receptor to elicit a signal. So we're going to have basically a receptor right here, and then the signal comes in, and once the signal reaches and sends the message, it's going to elicit a response inside the cell and say what should be done. And this is kind of like the idea of the lock and key. And this is specifically seen in immune cells. And if you think about it, you don't want any mistake in your immune cells because you don't want an autoimmune disorder. So this is a very selective process, a very regulated process to ensure that there is no misbinding or incorrect binding of the signal to the receptor, and illicit responses, incorrect responses are elicited. So we want to make sure the right response is elicited to ensure our health. And so if we think about it, typically what's happening in cell signaling, it functions by going through a cascade or process that is not simply just a one-step thing. So if we go back to our original idea of the signal, so we have a signaling molecule that has its own little nucleus. So here's our signaling cell, and it releases the signal. And so it's going to have something to transfer that signal to the desired location. So we're dealing with a hydrophilic signal that's going to go to the receptor and send that signal out that we desire. And now that we've detected, so we've had the signal come in, it's been detected, and now that signal is telling the inside of the cell, okay, do some job. So we have one option where the signal can go to the nucleus and allow for transcription, or the signal can remain outside of the nucleus and stay in the cytosol. Or it can continue down a cascade within the cytoplasm, or the cytosol, sorry. And so in this method, since there's a cascade, there's also amplification, because as I send the signal down, the next person that carries it has to have the stronger signal to make sure that the signal actually is processed correctly. So this is the general kind of view of how the signal has to go through. In the next videos, we're going to look at different types of receptors now that we have an idea of how the signal is sent to the receptor and inside, what the functions are, or what happens when it goes inside the cell. We're going to look at different types of receptors and see what their impact is, what they're going to do. If you like the video, please give it a thumbs up and subscribe to the channel.